Today we are going to talk about the criteria for classification, that is classification of bacteria according to Bergey's manual. So Bergey's manual is one of the standard classification method, and what are the criteria defined in the Bergey's manual is still continuing, and microbiologists following the same criteria to classify the microbial world of different class and different orders of different hierarchies based on the Bergey's manual. Uh, okay, as we discussed on the last turn about the importance of the classification. So the importance of the classification will be more clear when we detail discuss about the different reason why classification is important. That is defined in the Bergey's manual, and what are the different categories to be defined classification, and to keep these microorganisms on different level in hierarchies is again defined by the Bergey's manual. So Bergey's manual is very important, and one should learn it. How need not to learn each and every aspect of Bergey's manual to cram the whole Bergey's manual, but I think we should know the important parameters and different editions which are there. So sometimes the questions may ask on these edition of 1923, 1984 and so on. So today we'll talk about the same. So Bergey's manual, the authority on the bacterial classification. Bergey's was the authority on the bacterial classification, and in 1923, his name David Bergey and his colleagues, co-authors at the University of Pennsylvania, published the first comprehensive bacterial classification. So that was the first in 1923. And this manual, which is very determinative and detailed expression of the whole classification mode in bacteria for the benefit of the microbiology and the world of microbiology. In 1984, systematic editions came, and this systematic edition, first edition of Bergey's manual on systematic classification, on systematic bacterial classification, published, which describe all known prokaryotic species. So all the prokaryotic species which are so far until 1984 discovered were endorsed in the systematic edition. In 2001, almost 3 decades ago, the present edition, which is like continuing, we call the second edition. This edition is based on the phylogenetic approach adopted with the 5 volumes planned based on the molecular analysis. Now, if you recall my last lecture on the molecular identification of the classification, so now this edition, which is quite advanced edition compared to the other editions, in which the classification was based on the molecular aspects, and on the basis of the molecular aspects, they classify these bacteria or all the prokaryotic species into different classes and categories. Now, this was a revolutionary shift from phenetic to the phylogenetic. Now, what the phenetic means? Phenetic means earlier we talked about the morphological, physiological, and some phenotypic expression of the bacterial world or the microbial world, where on the basis of the phenotypic expression, on the basis of the size, shape, color, texture, right, and then culture characteristics as well, physiological responses of the bacteria as well, we categorize the microorganisms by enumerating the characters into like related and unrelated characters, but that was not on the basis of the evolutionary pattern or evolutionary background of these microorganisms. So there's a shift in this Bergey's manual where he defined the phenetic to phylogenetic changes. Now, phenetic change, as I just told, they're based on the observable, which are seen, which can be seen by naked eyes, features, characteristic features, and physical trait of the organisms. Whereas the phylogenetic classification is based on evolutionary relationship revealed through the molecular analysis. Their genotype, their GC content to some extent, their hybridization, their rDNA and so on. So basically the rDNA sequencing or the gene of ribosomal RNA gene sequencing is one of the most important method of classification of the species with the different genera and the groups. So this Bergey's manual is defined basically the shift from phenetic to phylogenetic analysis or phylogenetic method mode of classification, where one can very precisely and certainty, with the certainty, define a different bacterial species in this Bergey's manual. Now then comes the negative, gram-negative staining. Gram-negative and gram-positive staining are very common. You all must know it. So there's a bridging phenetic and phylogenetic classification by gram staining. So considering gram staining based on the phenotype as well as the phylogenetic analysis of these prokaryotic species, the Bergey is defined in his manual. So while gram staining properties generally considered as a phenetic characteristic features, they are also play a crucial role in the phylogenetic classification in terms of evolutionary changes which taking place in that. Now the fundamental differences between the gram-negative and gram-positive bacteria reflects the deep revolutionary shift from phenetic to phylogenetic, right? So this deep evolutionary divergence, so on the time scale, they evolved in more advanced version of these species by mutation, by changes, by variations. So it supports the molecular evidences, right? Supported by the molecular evidences. For example, their genotype, their gene content, their different other kind of characteristic features, which we call the genetic characteristic features, different markers. Now this makes gram staining one of the few classical techniques that remain relevant in the modern phylogenetic classification. Now when we come to gram-negative versus gram-positive, so that is the first line of differentiation between the prokaryotic species or prokaryotic whole family which belong to the gram-negative and which belong to gram-positive, and the fundamental differences. Now the cell wall structure, the gram-negative complex, so on the basis of different characters in the gram-negative and gram-positive, so cell wall structures, then metabolic diversity, and then structural features. On the basis of these characteristic features, we define the gram-negative and gram-positive. We compare these two different class of the bacteria. Sorry. The gram-negative complex wall, it has a complex wall with the peptidoglycan of 2 to 7 nanometer. An outer membrane is 7 to 8 nanometer containing the lipopolysaccharides. Now comparing to gram-negative, gram-positive has a thick homogeneous cell wall of 20 to 80 nanometer composed of mainly of peptidoglycan. So the content of peptidoglycan is more in gram-positive than gram-negative. So this is one of the different characters between these two, and we can differentiate gram-positive and gram-negative. With the teichoic acid is also present on the membrane. Now in metabolic diversity, the gram-negative highly diverse phototrophic bacteria, chemolithoautotrophic bacteria, chemoorganoheterotrophic bacteria. So these are some metabolic responses on the basis of the energy consumptions, on their food productions, and their survival on different states. So we categorize the gram-negative bacteria like that. Now, if you compare the gram-negative to gram-positive bacteria, usually they are the chemoorganoheterotroph, right? Chemoorganoheterotroph means they are getting the energy from chemicals, right? And they are dependent on some other food, right? Some organic food to get the energy for the survival. With the few phototrophic species. Phototrophic species means the light is required, and in the presence of light they can synthesize their food, or they can get their survival commodities, they can survive on that, or they can get the energy for the survival. Now if you come to the structure of gram-positive and gram-negative to compare these two classes of bacterial population, so gram-negative are the diverse appendages including the pili, fimbriae, prostheses and stalk, and they cannot form the endospore. So this is one of the difference between gram-positive and gram-negative. Endospore are not present in that. Now if you compare to the gram-positive, in gram-positive usually they lack the appendages, but some groups can form the endospore for the survival. Now if you remember, when we talk about the tyndallization, we discuss about the spore forming bacteria, sterilization of spore forming bacteria. So these are the same kind of bacteria, that is gram-positive, which may form endospore, and these endospores basically later on provide difficulty in sterilization because they reappear again after the sterilization. So we have to follow the tyndallization or Tyndall effect rather in order to sterilize such kind of a bacteria. So this is the basic difference between gram-positive and gram-negative. Now when we come to the different classification or the 5 volumes of these Bergey's manual. These 5 volumes of the Bergey's manual are based on the structure of a modern categorization basis of these Bergey's manual. Now, the second edition of Bergey's manual is organized into 5 comprehensive volumes, each focusing on specific phylogenetic groups. Now, when I say phylogenetic group, it means we talk about the evolutionary changes due to the mutation, due to the changes and variation. Now, this structure reflects our modern understanding of prokaryotic evolutions and provides a systematic approach to bacterial classification. Now, this is how the whole Bergey's manual is based on the advanced form of the Bergey's manual, the latest form of the Bergey's manuals. Unlike the first edition, where the pathogenic species are no longer grouped together but are placed according to their phylogenetic relationship. Now, the five bases of this Bergey's manual are categorization of these bacterial species are the archaea, deeply branching bacteria, second protobacteria, then low GC content gram positive bacteria, includes the firmicutes with a lower guanine cytosine content, and the high GC gram positive bacteria, that Acinetobacter are actinobacteria with high guanine cytosine ratio. Then diverse bacterial phyla are also present, where we have Planktomycetes, then we have spirochetes and other specialized group of bacteria. So these are the 5 different categories of bacteria which are present in the Bergey's manual to define detail. Now, let's talk about the volume 1. The ancient life form of these bacteria which cover in the volume 1 like archaea and the most ancient bacteria lineage, right? And these as I have already talked about the linkage between archaea and bacteria are eubacteria. There are some common features of the archaea and eubacteria, so that show the evolutionary relationship between the archaea and eubacteria. Now, these organisms represent some of the earliest form of life, like on Earth, and often thrive in extreme environment, that minor early Earth condition. The archaea, despite their prokaryotic structure, are as genetically distinct form of bacteria, right? As they are the eukaryotes, they are from eukaryotes and representing a unique domain of life. Now, the domain archaea and ancient bacteria, if you compare, you find the in domain archaea we have a Crenarchaeota, which are basically thermophiles, because mostly archaea bacteria or archaea domain, they have extremophiles, as we discussed on my last lecture. Then we have a Crenarchaeota, then we have a Eurochaeota, and Eurochaeota usually we have a methogens, which are methane producing bacteria, then halophiles, and extreme environmental the species of the bacteria are present. These extreme environmental specialist species are again under the category of archaea. So these are defined in the domain archaea. Now, in ancient bacteria, the deeply branching lineage, then we have a phototrophic bacteria, then we have a thermophile species. Although thermophile species are also part of the archaea domain, but I think they are also ancient, or earlier they are also found without defining any domain. Now, archaeal diversity, as we know, the thermoprote, then methanogen, then we have halophiles, and then we have hyperthermophiles. Now, these 4 categories of archaeal, which are very commonly found, the bacteria defined under the archaea domain under these categories are under these having these features or attributes. Thermoprote is basically the extreme thermophiles, where they can tolerate the high temperature, right? Thermoproteus and sulfolobus, which are also present in the sulfur containing bacteria, or they can also thrive in the high temperatures exceeding 80 degrees centigrade. Even their optimum temperature sometimes go to 70 plus. So it means they can even not only thrive and not only survive at high temperature, but they can have better growth at the high temperature up to 70-75 degrees centigrade. And they are mostly acidic conditions are required for that. Then methanogens are those which are methanobacteria. They are methanococci and methanomicrobia, and they basically produce the methane gas as a metabolic byproduct in anaerobic environmental conditions. So many times the methanogens are present in the conditions where they require no oxygen, and even in anaerobic condition they can produce the methane. Halophiles are those bacteria like halobacteria which require extremely high salt concentrations, and it means the lake which are salt lake, high alkaline lake, where the halophiles can survive, and often they give salt lakes, right? Their characteristic is pink colorations. Now hyperthermophiles, thermococci and archaeoglobi is basically represents some of the most heat resistant and organisms known to science. Basically the discovery of the PCR. I mean, this is not the part of this course, but I just to give you an example of PCR Thermophilus aquaticus, one of the thermophile. The enzyme isolated from Thermophilus aquaticus, we call DNA polymerase or Taq polymerase, was supposed to be one of the breakthrough for the PCR machine, a PCR reaction, and that was Kary Mullis got the Nobel Prize on the same discovery. In case of protobacteria, which is called powerhouse, and under the volume 2 they define the protobacteria. Protobacteria represent the largest and the most metabolically diverse group of bacteria. So there are a lot of metabolic changes taking place in protobacteria, and this phylum encompasses an extremely range of lifestyle, from photosynthetic purple bacteria to up to pathogenic species, right, like Escherichia coli, Salmonella, and so on. The 5 classes within the protobacteria demonstrates the remarkable evolutionary success of this group across the virtually even ecological niche on the earth. Now, alpha protobacteria, beta protobacteria, gamma protobacteria, then we have a delta and epsilon protobacteria. Now, these are the different classes of the protobacteria which are defined in this Bergey's manual to more comprehensively and more detailed. It includes the alpha protobacteria, includes the nitrogen fixing like Rhizobium, intracellular, Rickettsia, and stalked colobacters. So these are the categories of the alpha protobacteria. Similarly, beta protobacteria, we have contain the Neisseria, gonorrhea causing bacteria, pathogens, and ammonia oxidizing Nitrosomonas, which are very common bacteria present in the soil, symbiosis with the plant, especially leguminous plants. Then gamma protobacteria are largest class, includes the Escherichia coli, Pseudomonas, and some enterobacterial member of the family of the bacteria, and many enteric bacteria, like some bacteria which are present in the intestine. In delta protobacteria, the sulfate reducing bacteria and the predatory Vibrio bacteria, which are also belong to some other class of Vibrio class, where the cholera causing bacteria, they have some pathogenic characters also. Now, in epsilon protobacteria, the spiral shaped bacteria like Helicobacter pylori, then Helicobacter other species, and Campylobacters. Now, look at the beauty of this Bergey's manual, especially volume 2. They have defined the protobacteria in a very nicely, and all categories of the prokaryotic species are defined on the basis of the different characters, both phenetic and phylogenetic characters, which make them, on different branches in the class of the protobacteria. Now, in volume 3, they define the low GC content gram-positive bacteria, mostly firmicutes with low GC positive bacteria, includes some of the most industrially, medically important organisms, which have a lot of application in both industrial applications and clinical and medical applications. And this group is characterized by a relatively low guanine and cytosine content. I mean, this has some other logic which probably need another half an hour to talk about on this, but I'm not going to talk on this reason. But I think just to know what the GC content, low GC content is as one of the characteristic feature of this firmicutes. Well, now it has 3 basically classes we call Clostridia, which are anaerobic spore formers, includes the Clostridium species, Clostridium botulinum, Clostridium, there are many other. They are highly toxin bacteria. They produce a toxin like Clostridium botulinum, which is producing the botulinum toxin. Then they have also tetanus toxins and so on. Some producing the deadly toxins, endotoxins, and they are very, very deadly because even very, very small quantity of these toxins can harm the big populations of the human and animals. The molecules are the wall-less bacteria, while the mycoplasma, representing the smallest free living organism with minimal genome. So this is another very nice class of bacteria. Then bacilli. Bacilli is referred to as a rod-shaped bacteria. Include the bacillus, lactobacillus, streptococcus, then staphylococcus. They're all gram-positive bacteria and crucial for the food producing are the bacteria and human health. They're good for the human health. Now, in volume 4, Bergey defined the high GC content bacteria, gram-positive bacteria. They are the actinobacteria, actinomycetes, for high GC gram-positive bacteria. It represents one of the most important, most economically important group of microorganisms because they are used for various industrial applications. They are used for various medical applications, and these species exhibit the very complex development cycle of filamentous growth patterns. Now, antibiotic producers, Streptomyces species are the pharmaceutical industry's workhouses, right? Because they are medically important, they produce the various antibiotics and several antimicrobial agents, right? Tetracycline, then streptomycin, and various other life-saving molecules are produced by these producers, are gram-positive bacteria. Now, mycobacteria include the Mycobacterium tuberculosis, which is one of the most common bacteria causing TB, very common disease and very prevalent in our country. Then Mycobacterium leprae, which is causing the leprosy, which is again very commonly found in India and subcontinents. These bacteria causing the tuberculosis and leprosy, they have a very unique waxy cell wall. So they can be characterized and identified through phenotypic expression of having the very unique waxy cell wall. Now, industrial application of these gram-positive, high GC content gram-positive bacteria. Now, when I refer high GC content and low GC content, you can guess that high GC content could have the genome could have high temperature required to denature. COT value is high. Low GC content means it could have a COT value is low. So what does the COT low and high means? Complexity of the genome. Well, the complexity of the genome is again another area to discuss, which again I'm not going to talk about the complexity of the genome, but just to refer the GC content may refer to the complexity of the genome. Now, industrial application is cyanobacterium. Then we have a propionibacterium. Then we have several species of these genera used in the amino acid productions, some protein productions, and food processing industries. Their enzymes are very commonly found in that, and they also produce certain enzyme which can metabolize these production of these amino acids, both different classes of the amino acids, aliphatic amino acids, aromatic amino acids, branch and unbranched amino acids. Now, volume 5 is a specialized and diverse bacterial phyla. In volume 5, they encompasses the remaining bacteria which are not included in the 1, 2, 3, 4, 5 volumes. Major bacteria we have already discussed in 1 to 4 volumes. And in this phyla, they don't fit into the previous categories. All bacteria were fit in the volume 5 in the Bergey's manual. This diverse collection includes some of the most unique and specialized bacteria from the spiral-shaped spirochetes and causes the syphilis, very common sexually transmitted diseases, then Lyme diseases and intracellular chlamydia, chlamydomonas that are obligate parasites. Now, these groups demonstrate the incredible diversity of bacterial life from the ecological strategies. I mean, they have different niches. They are found in different ecological states. Now, if you talk about the spirochetes, the masters of the motility, call it, because spirochetes represent one of the most distinctive bacterial morphologies that have like different characteristic features, right? Have different shape, screw shape, cork screw shape, Like this, we have, this is kind of a shape we have. Then we have some unique features of these motility. They can move on the surface, on the plate, on the media. The bacteria move through viscous environment, then using axial filaments. So they have also endoflagella, which is part of the bacteria. For example, this is the bacteria. Now this bacteria may have some flagella like this, and this flagellar movement can help the bacteria to move on. Now that run along with the length of the cell bodies. For example, we talk about the Treponema palladium, Borrelia burgdorferi. The Treponema species are causative agent of syphilis, a sexually transmitted disease or infection with the complex clinical stages, and it may be deadly if not properly cured at early stage. Borrelia burgdorferi is transmitted by tick, cause Lyme diseases with the characteristic bull's eye rashes and so on. Then leptospira, the waterborne diseases we call waterborne pathogens causing the leptosporiasis, and then often contracted through the contaminated water. So these are some features of the Spirochetes pathogenic bacteria which are considered under the Bergey's manual. Now chlamydia is obligate intracellular parasite. Now chlamydia parasite is represent the unique evolutionary strategy. It has obligate intracellular parasite. So there's a difference between intracellular and extracellular parasite. So this is the class belong to the intracellular parasite. Now the bacteria have evolved into complexity dependent on the host cell for survival and reproduction. So because it is a parasite, it depends on the host. So what are the host? Right, the specific host provide the survival or support to survive and to produce. That's how the Koch postulate, as I have discussed in my first lecture, probably first or second lecture, we will talk about the Robert Koch postulate, how this infection is established. Now, they undergo a complex development cycle alternatively between infection, elementary bodies, and replicative reticulate bodies. Now, chlamydia species are major cause of sexually transmitted infection and trachoma, that leading the infection cause of blindness worldwide. It has 4 different categories: elementary bodies, attachment and entry. Then elementary bodies like infection metabolically inactive that survives outside the cell. So it survives outside the cell. Then attachment and entry. So for second step is entry, elementary bodies attach and enter into the host cells and then replicate, and then reticulate body transform into the metabolically active replicate. So these are the 4 categories, the 4 steps of this infection, where it attach first and then it enter inside and then replicate on the host machinery, and then it moves on and to cause infection by spreading. The unofficial nature of bacterial classification, right? This candid statement from the Bergey's manual edition reveals an important truth about the bacterial taxonomy. That is, there is no such thing as an official classification. So it means official classification means like no classification is a standard and static. It could have changed on the basis of time scale, and you can add some more characteristic of a molecules to define them, to further characterize, and so on and so forth. It is merely the best consensus at the time with the scientists, with the researchers, taxonomists, and balanced view that there are also always a region in which these organisms have been categorized or have been found. So it depends on timeline, it depends on the region, it depends on the peoples who categorize them, or taxonomists who categorize them. So you can have some kind of a stable classification method. Now, I think this is all done with this lecture. So now we have another lecture soon start on another topic after this. Thank you very much.